So I like the conversation about the planes. So I had a very long flight from the UK to the US. And when people ask me on the plane what am I doing, I tell them I work on plants in the ocean. But then we end up talking about whales. So I suppose whales are the dogs of the ocean. I'm not sure, but you know. Everybody knows whales, so I have a lot of conversation about whales. Although I don't work on them, but I have actually one of them in my presentation. So before I'm going to talk about the evolution of diverged alleles in diatoms, which basically will be a presentation about the genome project of Fragileiopsis cylindrus, which lives in the Southern Ocean, I'm going to acknowledge my collaborators. And first and foremost, it is JGI, because the project was funded by JGI many years ago. I think it was 2008. And it came through the CSP program, and I'm very grateful to Igor, Bobby, Asaf, Jeremy, Carrie, and many others. So I had about 40 groups involved from many different countries to help me to annotate the genome and to understand what's going on in this very weird organism, because the genome is very different to anything that had been sequenced before, especially when we compared it to the other diatom genomes that were available at that time, like Thalassosaurus, Pseudonana, and Fragileiopsis. I'm also Pheodactylum, and I'm also very grateful to my group. Jan Strauss did a lot of annotation work of Fragileiopsis, and he also contributed with EST libraries and looking at differential expression. Then we had also some sequencing done in Norwich at the Earlham Institute. So we did PacBio sequencing in the group of Matt Clark. Pirita was leading this. And then I had some population geneticists helping me to understand how the diverged alleles evolved over the last 100,000 years or so. So this was Kok van Oosterhout, Mark McMullen, and Ben Ward. And then we did not only have funding from JGI, Department of Energy, but also from other funding resources, because it took quite long, really, to publish this. So I start very, very basically, where do marine diatoms live? And this is an animation here showing the 4 most abundant phytoplankton groups in the ocean. It's very simplistic, but it helps to bring across a very important message here. So diatoms are in red, and what you see, they don't like warm water, okay? So they don't live, or they don't dominate phytoplankton communities in the tropics or subtropics. They prefer to live here in the North Pacific, North Atlantic, and in the Southern Ocean, specifically in the Southern Ocean, because the water there is cold with a lot of nutrients, and then they form blooms and bust. And this is what you see here currently. So the spring blooms take place in the North Pacific. This runs through several years and seasons. And here we have diatoms blooming now in the Southern Ocean over the spring and the short period of summer. So in other words, these organisms are very significant for the global carbon cycle. So about 20% of annual carbon fixation is due to the activity of diatoms. So the oxygen for every 5th breath you take comes from these organisms. And if you put all the phytoplankton groups together, then they're as important as plants on land, okay? So this is enough reason to study them. So let's zoom in a little bit into the Southern Ocean. And this is another animation of Mick Follows. This shows the temperature variation in the surface of the Southern Ocean. It's a pretty cold ocean. It's the largest cold water ecosystem on the planet. In the middle is the Antarctic continent, very much isolated by these cold water masses. It's like a merry-go-round ecosystem. They circulate around the Antarctic continent. And by looking at this, you get an impression how isolated the organisms are that live in there. And they had to come up with very specific adaptations in order to cope with low temperature and also the strong seasonality. And these organisms form also very unique food webs. And this is my slide where I show a whale. And this is one of the shortest food webs. I wouldn't even call it a food web. It's a food chain consisting of 3 different trophical levels. One are the diatoms. They're really the most important primary producers in the Southern Ocean. Then we have krill, and then we have whales. So very efficient transport of energy and carbon. Krill, by the way, is considered to be the most successful animal on the planet, believe it or not. So the population consists of about 700 trillion individuals. Can you imagine? So we have about 8 billion human beings on the planet. So this population consists of 700 trillion organisms. They link, this is a keystone species in the Southern Ocean, they link the lower trophical levels with the higher trophical levels. And although this organism is so important, no genome sequence is available for krill. And as far as I know, there's not even a project underway to try to sequence this genome. So this is something to think about. Another very interesting feature of the Southern Ocean is its biogeochemistry. It's very rich in macronutrients, and this is what is shown here. So you see the nitrate concentration here, and the concentration goes up to 30 micromoles in the Southern Ocean. This is very unique, but it's not only nitrate, it's a silicate and phosphate as well. And you have not many other oceans or not many other areas in other oceans that have so high nutrient concentration. There's an area here in the North Pacific, and the equatorial Pacific has also some elevated nutrient concentration. Why is that? These areas are also called HNLC areas, and HNLC stands for high nutrient, low chlorophyll areas. And the reason why there are high nutrients, although there is phytoplankton in these areas is limited by dissolved iron. Yeah, so they can't benefit from these high concentration of macronutrients because their growth is limited by dissolved iron. And this is very, very, very much the case in the Southern Ocean. I'm afraid all this, and this is getting now a little bit depressing, all this is threatened, as you know, and I'm sure you are well aware of these data, is threatened by global warming. So we pump too much CO2 in the atmosphere, and this heats up the planet. And most affected are the northern hemisphere of our planet, and specifically the Arctic Ocean. So there's a lot of sea ice retreat in the Arctic Ocean, which affects the entire ecosystem. And when you look at the bottom of Earth, where the Southern Ocean is, and it's still considered a very pristine environment, but we still see a heating up of the Southern Ocean, of the Antarctic continent, especially here when you look at the peninsula. We see that over the last about 50 years, the temperatures have risen, and now shelf ice breaks off and the big icebergs drift around, and then eventually they melt in the Southern Ocean. Before we can make any assumptions about how organisms cope with global warming, I think we need to put a strong emphasis on trying to understand how organisms have evolved under the current environmental conditions. And this is why we selected the sea ice ecosystem. Sea ice covers, it's very unique for polar ecosystems. Sea ice covers about 10% of the Earth's surface, making it a major biome like all tropical rainforests combined. And when you go with an icebreaker, either to the Antarctic or to the Arctic Ocean, then you break up the ice cover, right? And what you see is what I show here. So this is an ice flow turned upside down. So you look actually at the underside of the ice, and the underside is covered by dense populations of diatoms. And they are really the feeding ground for juvenile krill. So they come up and then they feed on these ice algae. So when all this disappears, the 700 trillion krill species or individuals will also disappear because they're very much dependent on these sea ice algae. This is a very extreme environment. This image speaks for itself. Temperatures are very low. They're at the freezing point of seawater. We have very restricted supply of nutrients because this is a semi-enclosed habitat. These organisms live within brine channels, and also the light intensity is not very high because usually there is snow cover on top of sea ice, and snow reflects a lot of light, so they have to be adapted to be able to cope with very, very low light intensity. And then, of course, the salinity is also relatively high, so up to 10 times as high as seawater. But these organisms are around, they're not only coping, they thrive. Right? And we were interested in specific adaptations and what they evolved over the last millions of years in order to be able to cope with these extreme and very unique environmental conditions. And Fragileiopsis cylindrus is one of the key phytoplankton species, not only in the Southern Ocean. It's actually a bipolar species. We are also very keen to sequence the Arctic counterpart. We have not done this yet, but we plan to do this. But this is an isolate from the Southern Ocean. It is not only abundant in Antarctic sea ice, but also at the ice edge zone. It's a bloom former, like what I showed in McFollo's animation. It forms large blooms when the conditions are good. And it's a psychrophile, so it can't grow above 12 degrees centigrade. It was also the first genome from eukaryotic psychrophile. So when the genome sequence became available, we looked at metal-binding proteins because of the interesting biogeochemistry of the Southern Ocean. Phytoplankton, as I said before, is limited by iron. So we looked at iron-binding proteins, copper-binding proteins, and zinc-binding proteins. And what I show here in the upper part are iron-binding proteins for several different organisms listed here. And with the green star, you see Fragileiopsis cylindrus. So there is not really an enrichment of metal-binding proteins in the genome of F. cylindrus. But when you look at the copper-binding proteins, then you see a slight enrichment. And this was known before based on very targeted studies with phytoplankton from these HNLC regions. So most of the electron carriers in photosynthesis, they contain iron as a cofactor. And organisms from HNLC, so phytoplankton from HNLC regions, they tend to replace those electron carriers with iron as a cofactor with electron carriers that have copper as a cofactor, because the copper concentrations in these ecosystems are slightly higher than the iron concentrations, and this makes them more competitive, right? What came as a surprise to us is shown here at the bottom under D, an expansion of the zinc-binding protein families. We didn't expect that. You see here again Fragileiopsis, and then you see other organisms, the other diatoms. We sequenced Emiliania huxleyi, chlorophytes, and so on. Significant enrichment of zinc-binding proteins. So why took this place in this organism? We have no clue yet. But maybe one clue comes from the biogeochemistry, again, of the Southern Ocean, and I selected an image from a recent paper published by Vance in Nature Geosciences that shows that the Southern Ocean basically is the ocean with the highest zinc concentration. There's no ocean that has a higher zinc concentration. So maybe this is why they benefited from the high zinc concentration in the water and therefore expanded the MUNT binding, specifically the MUNT zinc binding proteins. MUNT proteins are very often involved in gene regulation and regulation of transcription, so maybe this contributed to the adaptation of this organism to this extreme environment. This is pure speculation. We have no evidence for that, but we certainly will follow up on this. So the zinc concentrations are very high, as I said, in the Southern Ocean, and when you approach the polar front, this is PF, this is Antarctic divergence, this is polar front, then the concentrations drop. And the same is the case for other macronutrients. So they really drop down, and when you leave the Southern Ocean, the concentrations are really low in general. But one of the most surprising results, really, and it took us a long time, I can say that indeed it took us a long time to understand what's going on, was the discovery of the allelic divergence in this genome. And as I said, this has not been discovered in any algal genome before, and I'm not aware this is present in a lot of plant genomes. Please correct me if I'm wrong. But here are some general genome statistics, and this is based on the two haplotypes, so diatoms quite often are diploids. And the genome size based on the single haplotype is about 60 megabases for F. cylindrus. And Phaeodactylum and Thalassosauria, the genomes are a little smaller, but the increased genome size in F. cylindrus is largely due to repeats. 35% of the genome are repeats, and these repeats also include transposable elements. When we look at the predicted genes, we found that about 6,000 of them are diverged gene pairs, alleles, which means about 28% of all gene pairs in the genome. About 56% of them have a nucleotide identity of about 90%, and many polymorphisms between homologous scaffolds are not genic. So the polymorphisms can be up to 6%, which is moderate to high for any organism, but we find most of them between genes, but also in regulatory parts. In the interest of time, I can't talk about this, but we found a lot of mutations in the promoter regions, which is interesting when it comes to gene regulation, right? So the question we had to address was, why have these alleles really diversified in this genome? And we had two hypotheses. The how maybe is due to the accumulation of mutations, and the why maybe is due to the adaptation to the very variable and extreme, as I showed before, environmental conditions. If both would be right and true, the diverged alleles would be the result of diversifying selection. As I think no one or no talk has addressed the theme of diversifying selection, I just briefly introduce that and remind you what diversifying selection is. Diversifying selection can also be called positive selection or disruptive selection, and it describes changes in population genetics in which extreme values for a trait are favored over intermediate values. And this can lead to speciation. I've picked the rabbits because it's very easy to explain with this example, and we stay in the theme of polar science. So in polar regions, white rabbits are favored by selection, right? Because they blend very nicely into the environment. So predators like foxes or polar bears have really difficulties to catch them. But if snow disappears due to global warming, the population will change towards extreme traits. And we have the gray rabbits on one side, and we have the gray and white rabbits on the other side. So changing environments can change populations in different ways. And we were interested as to whether we find something like that in Fragilaiopsis cylindrus, and whether this was also driven by the environment. How to study diversifying selection? You need to measure the NDS. This is the ratio of non-synonymous over synonymous substitutions. And when you have data that are at 1 and higher, this is indicative for having positive selection. And if your results are lower than 1, this can indicate either purifying selection or neutral selection. And most of the mutations are not beneficial at all, so they are either under purifying selection or neutral selection. So coming back to F. cylindrus. We had to test, first and foremost, the accumulation of mutations, and this was a bit difficult because what we found was really not found in any algal species before, and we wanted to compare like with like, meaning alleles with alleles. But we were lucky, a group in Naples, and this was also funded by JGI, they sequenced mate pairs of a closely related diatom, Pseudonychia multistriata. It's not a polar species, it grows in the Mediterranean, but then we used our diverged alleles from F. cylindrus, and we compared them to homologs from Pseudonychia multistriata. And this is what I show here in this graph. You have on the y-axis a DS, so these are the synonymous nucleotide polymorphisms, the S of FC, and here on the x-axis you have the S for Pseudonychia multistriata. And what you see, cylindrus has accumulated much more mutations for these diverged alleles in comparison to Nichia multistriata, about 8 times higher DS in F. cylindrus compared to PM. This figure shows synonymous nucleotide substitutions per synonymous site, expressed as Ks. In black, we have Pseudonychia. In gray, we have Fragilaiopsis. For recombining organisms, Pseudonychia multistriata has frequent sex on a regular basis. For recombining organisms, you usually see an L-shaped curve. Most of the sequences have a very low number of mutations, and some have higher mutations. Ks is here on the x-axis, and this is just a frequency. But for Fragilaiopsis, for the diverged alleles, we saw almost a perfect bell-shaped curve, with many more sequences with a lot of mutations and only a small number with a very small number of mutations. And you can use the amount of mutations to date the sequences. So on this end, we have very old sequences because they had a lot of time to accumulate mutations. And on this end, we have very young sequences that didn't have a lot of time to accumulate a lot of mutations. And when you take this into account, into consideration, then you can say here in the middle, this was the time where the majority of these alleles diverged. And we were very interested to replace Ks with years. And we did that, but there is not much known about these organisms because they are extremophiles and so on, so we had to apply some assumptions. And one assumption was a constant mutation rate of 10 to the minus 9 mutations per base pair and per generations. We assumed a division rate of 0.1 divisions per day, and we assumed a growing season of 4 months per year, with each month consisted of about 30 days. And with all these assumptions, we replaced Ks with the coalescence time between two alleles, shown here at the x-axis. And we found that most of the alleles here diverged about 100,000 years ago. And the blue curve basically represents the gray curve in this figure. But what are the green curve and red curves are about? So the green curve, we looked into natural populations as well. So the green curve is a ribosomal protein we amplified from natural Fragileiopsis populations. And then we use Sanger sequence to get very high accuracy of the reads in order to do the same calculations. And the red curve is an iron transporter gene, also amplified from natural populations. And all of them overlap very, very nicely. And this was very reassuring. This told us what we see in a model species is really also happening out there in the environment. So why 100,000 years? So we thought about this, we looked in the geological record and so on, and then we found a coincidence. About 100,000 years ago, this was the onset of the last ice age on Earth. And this is shown here in this figure where you have the yellow parts as the warm phases and where you have the blue parts as the cold phases. And here's 100,000 years. So this was the onset of the last ice age. But this is pretty puzzling because Fragileiopsis lives in the Southern Ocean at least for a couple million years. Why have they started to develop these diverged alleles only 100,000 years ago? We have no answer to this question, but this is a very interesting coincidence. What we also saw, we did RNA Seek, as I said before, done by Jan Strauss. And we did this for the whole genome, but what I'm showing here is only the set of diverged alleles, the 6,000 genes or so. And we did this for important environmental conditions, low iron, low temperature, high CO2, and darkness. And you have here, for each of the treatment, and all this is in relation to happy growth, you have for one column allele set 1, and for the other column allele set 2. Darkness certainly had the largest effect. This makes sense. This is an autotrophic organism. If you switch off the light, then you change the metabolism a lot. But not everything is downregulated. Also, a lot is upregulated. But then if you look more closely and compare the different sets of alleles, you see very big differences. For instance, here at elevated CO2, there's a cluster downregulated for one set of the alleles, but the other set is not differentially expressed at all. And then here you have differences just in the opposite directions. You have downregulation for one set of the alleles and upregulation for the other set of the alleles. So this is very, very interesting. And we also looked at Go enrichment, and this just shows cold stress versus control and iron limitation versus control. Here, allele set 1 on the left, allele set 2 on the right. No need to identify all the details, but look at the big picture, look at the patterns and the differences between allele set 1. So whenever there's a large circle, red or orange, it means the Go term is very much enriched. So you see differences in the pattern between allele set 1 and allele set 2. So meaning that if you turn on one set and not the other, you change your metabolism. So you use your different sets of alleles to play and to adjust metabolism. The last question we had to address was whether the allelic pairs are really under diversifying selection. And this brings me back to the rabbits. So what you see here in this graph is on the y-axis, this is the maximum log 2-fold change between allele set 1 and allele set 2. What you see on the x-axis is the medium DNDS. And there's a positive correlation. So the higher the differential expression between the two allele sets, the higher is the NDS. And as far as I know, this has not been shown before, ever. Because usually the genes that are under positive selection are not as highly expressed as the more conserved genes. So this was a very nice result, which told us diversifying selection is driving the allelic divergence. And then we looked also at metatranscriptomes in natural communities, and we saw also biallelic expression. So 60% of the metatranscriptome sequence with homology of F cylindros match to the allelic variant pairs, although they only contribute to 28% of genes in the genome. So there's a bias It means they are really required to cope with environmental conditions. And in this semantic plot here, you see, whenever you see large circles that are red, there's only one allele present. And when you see blue circles that are very small, we were able to identify both alleles in the environment. So very strong biallelic expression also in natural communities. To conclude my talk, and this is an animation from NASA, and I hope this animation gives you some impression of how variable and how extreme this environment really is. It's a very cold environment. You have very strong winds that surround the Antarctic continent, and these winds, they induce a lot of mixing of the surface ocean. Sometimes the mixing can be down to 60, 70 meters. And then, due to seasonal temperature changes, you have sea ice formations. So a lid is being put on the surface of the ocean, which imposes a lot of stress on all microbes that live there. So the question is, this extreme environment with all these very variable environmental conditions, what is required to be successful in an environment like this? And I have a very simple answer to that, an allele for every occasion. And this has been evolved in Fragileiopsis cylindrus, but whether this is sufficient to cope with global warming, I'm not sure about, but we work on this. So I finish here. Thank you for your attention, and I'm happy to answer any questions you might have.
Thank you very much, Thomas. Questions?
Yeah, hello.
Who said that?
Where?
Stand up. Did someone have a? Yes. Okay.
That was great, Thomas. What I was going to ask you is that algae have different ways to deal
There's a mic behind you.
Algae have different ways to deal with extreme environments and fluctuating environments. One of the ones we found looking at extremophiles and hot springs is that they use a lot of alternative splicing. So they generate variation from a genome not by amplifying the alleles, especially if they're haploid, but they actually have a way of generating lots of novel exon combinations through alternative splicing under these stressful environments. Did you find evidence for that?
Yeah.
Fragileiopsis for this sort of phenomenon?
Yeah, a lot of alternative splicing. Some genes, they retain their introns. We have splicing at the 5' end of transcripts a lot. There are alternative start codons, and they make use of these alternative start codons. There's a lot of alternative splicing, even within the group of the diverged alleles.
So did that alternative splicing somehow have any meaning in the environment? Were you able to find that different environmental conditions led to different sorts of splicing products being created?
Yeah, it's a very good point. We haven't looked at this. This was very overwhelming for us to understand, to get a head around of how they regulate these diverged alleles. And on top of that, as you suggested, you have alternative splicing. So we need to dissect this a little more in order to understand the role of alternative splicing in addition to what we have seen here.
Question over here in the front.
Hello,
Yeah.
that was a nice talk. Fragileiopsis cylindricus isn't, it's in the Arctic, but it's not super common.
Yeah.
And the other interesting thing about it is one of the few species that actually does live in the ice and can also live in the pelagic zone. And so it's like that seems to be the main driver that would explain most of the alternate expression, I would think. And it would be interesting to see what happens in the Arctic where you don't have those species that are living in the sea ice in the water column. It's a total separation of species.
Yes, that's right. Melisara arctica is one example, right? It lives attached to the ice, but not inside the ice. Yeah, but Fragileiopsis, as you say, it lives in both sea ice and sea water.
Yeah, but that's only true for cylindricus. There's lots of Fragileiopsis that don't live in both in the Arctic as
like
well.
Kerguelensis.
Yeah.
Yes, right, yeah.
So I was just thinking about the zinc and the zinc connection. As you know, the zinc is also an important element for carbonic anhydrase. So can you connect eventually the CO2 concentrations to the physiology and to the apparent increase of zinc uptake in these?
Yes, so these diatoms have CCMs, and there are a lot of carboanhydrases encoded in the genome. They are very active, and especially in the sea ice environment, which is a semi-enclosed environment, you have very high pH values sometimes, up to 10, but they can cope. They use bicarbonate. And then generate CO2 by carbonic anhydrases. Yeah.
Yes.
Hi. So I'm not sure if you have the projections for it, but since genetic drift might be more increased as population size goes down, has there been like decrease in the amount of diatoms over the last 100,000 years that might explain why you're getting this increase in mutant allele frequency?
As far as we can tell, this is not the case for the Southern Ocean. So the Southern Ocean has not changed so much in terms of biogeochemistry yet, but we feel the heat in the Southern Ocean as well. And if the heating continues, then we might see changes not only in the population structure of species, but also in the abundance of different species. We see in the Arctic, we see invasion of temperate species going into the Arctic, and the polar species retreat.
Question. Do I understand correctly that the lack of this L-shaped distribution in divergence means that there's been no recombination for 100,000 years?
Yeah, good point. We looked at this very closely, and there are signs of recombination, but our estimates suggest that the rate of recombination is only about 4 to 5 times higher than the mutation rate.
I see.
And there is no indication of recombination within the group of diverged alleles. So they are excluded from recombination.
And do they have a particular position in the chromosome relative to the
No, they're
centromere?
scattered all over the chromosome.
Okay, but they're maybe prevented from recombining perhaps because of inversions or
Yes,
some
yes.
lack of hotspots or something like
Yes,
that.
yes, that's right.
Thanks.
Yeah, yeah.
Well, if there are no more questions, we have a caffeine break for the next half hour, and or not, whatever you choose. And we'll meet back here in half an hour. Thanks.